Great. Good morning.
I'll mute myself now.
Can everybody hear me? Amy, is everything good? So good morning, everybody. Thank you so much for the introduction, and thank you all for coming today. I'm really excited to be talking to you today about microbiome analysis and how we can apply that to beluga conservation. So in case you did not know, there are millions of microbes that live on and within us. And some of these are native, so they're there all the time, and those are known as the normal flora, where some of those can be introduced, and so those are the opportunistic microbes. And so the term microbiome really refers to all of the genetic material from all of the microbes that are associated with a host. And that term has really become very generalized, but it specifically refers to the bacterial communities of the host. So that is the true definition of microbiome, because there are other iomes such as viromes, so the viral communities, and mycobiomes, or the fungal communities that may occupy a host. And so all of these really play an important role in health. So microbiomes have been in the news for at least a decade now, but most of what we know comes from human research. We know that microbiomes play a role in diseases such as Alzheimer's and Parkinson's. And here's just a slide showing some of the way that microbiome research is impacting our lives, such as cookbooks that are feeding our healthy microbiomes in our guts, face creams that can treat and clear skins, and other things. But however, marine mammal microbiomes, including beluga whale microbiomes, are really unknown. And that is one of the focus of our research group. And as I mentioned before, the microbiomes can be composed from organisms from every branch from the tree of life, including the prokaryotes, or the true bacteria, eukaryotes, or the fungi or protists, and there's also another group called archaea, which are kind of like ancient bacteria. And then there are the viruses that are kind of their own different, whole separate thing. So for this purpose, we will stick to the traditional microbiome, and we will refer to that as those bacterial communities. So bacteria, they don't contain a nucleus, and they come in different shapes. As I mentioned, there are archaea, which are these ancient bacteria. They're closely related to, and they are very weirdly shaped, and a lot of them are extremophiles. So they're found in things like hydrothermal vents or things with very high or low pHs, so extreme environments. There's the eukaryotes, or things that have a true nucleus. Those can be single or multi-celled organisms, and they come in a wide variety of shapes. And then there are the viruses, and those actually need a host to be able to reproduce and survive. So we'll focus on bacteria for this talk, and bacteria have different shapes or morphology. So bacteria can either be rod-shaped or bacillus. They can be spherical or coccus-shaped, or they can be corkscrew-shaped or spirochete. Bacteria, some of them are non-modal, so they occupy a space and they just stay there. So those are non-modal, but other bacteria do have display motility. We have some bacteria here that have flagella, where they swim toward a target using a structure that helps propel them in their environment. Some bacteria are gliders. They excrete a goo out of their bodies, like through a jet propulsion motion, and then they can just glide across in that move from one to another. In the spirochetes, they use a corkscrew shaped motion. And so this is like the causative agent of Lyme disease, Borrelia burgdorferi, that has to move through that thick serum or plasma. So there are different microbial hotspots of an animal, and these are distinct and separate communities of microbes that inhabit specific sites on the body. So bacteria can take advantage of different niches and inhabit and start to occupy that. And some of these hotspots could be the oral cavity, the urogenital tract, the gastrointestinal or GI tract, as well as the skin. It is something to note and unusual about skin of marine animals is that their skin microbiome is distinct from the environment microbiome that they're living in. So the ocean water has its own microbiome, and that is very distinct and different from the skin microbes that are actually living on these animals. And then there is there is the respiratory tract microbiome. So the respiratory tract is the primary defender against inhaled pathogens. And it's lined with cilia or finger-like projections that helps trap foreign particles. And the respiratory tract is also inhabited by bacteria that forms biofilms. So it forms this protective shield that also helps protect the organism from invading pathogens. So beluga whales, like other whales, they breathe through their blowhole and not their mouth. So when they come up to the surface, the whale opens up its blowhole. They force warm air from their lungs out of their blowhole into the colder air. And this temperature change triggers the water vapor in the whale's breath to condensate into water droplets. So this is what happens when you go outside on a cold day and you can see your own breath. And so this blow contains millions of microbes. And these microbes hold important information regarding beluga health. But how do we collect them? Well, here at the aquarium, we are developing ways to capture those microbes from the beluga blow. And we can take advantage from the animals here at our aquarium that really do serve as ambassadors for their species. We can behaviorally train our belugas through positive reinforcement to voluntarily participate into various research behaviors, like blowing into a petri dish or into an inverted set of tubes where we can go ahead and capture that breath. In the wild, some researchers are utilizing drone technology like this snot bot pictured here that's used to collect blow from the whales. However, most of these studies are using large cetaceans, as you can see here from this drone flying through a plume from an Atlantic right whale. Those plumes are much larger than what a beluga would produce, which makes collecting breath from beluga whales a little bit more challenging than it is from larger cetaceans. We have been successful collecting beluga breath in the wild. We can utilize a pole and attach a petri dish to the end and stick it off of the end of a boat and wait for a whale to come up and surface to breathe, and we can capture that breath on that petri dish. In our lab, we are fortunate enough to have a master's student whose name is Elle Parks, who is currently working on a prototype for collecting beluga blow using a drone. Attached to the drone is a hula hoop type apparatus that's aligned with a material that would catch the breath of the beluga and hopefully the microbiome, the microbes that are contained within it. As I mentioned, that is challenging, and we are currently in the process of determining how low can we go next to a beluga to safely be able to collect this material. So the end goal of sampling this material is to perform or carry out what's called next generation sequencing. Next generation sequencing takes advantage of the unique properties of bacteria. So bacteria have their own ribosomes, which are distinct from the ribosomes that would be found in mammals. And we can go ahead and sequence that gene to detect what bacteria may be in a sample. And so we can go ahead and we can analyze a complete microbial community in a sample without ever even culturing any of these microbes in the lab. And that's very fortunate because at least 90% of microbes are not able or have never been cultured in a lab. So this allows us to find out what's in a sample and study really difficult microbiomes that would otherwise be impossible to determine exactly who is in there. The first step after we collect that is to extract the DNA or the genetic material out of the sample. So we presumably have captured some bacteria. Now we want to break those cells open and we want to get out their DNA. And we use that DNA as a target for PCR or polymerase chain reaction. And so using the DNA template and primers, which are little bits of DNA that are specific to the portion of the gene that we want to look at, we can mix that together in a tube along with our nucleotides, the Taq polymerase, which actually is going to do the work for us. And we're going to put it into a machine called a thermocycler. And in the thermocycler, in this tube, what is happening is the sample is first heated up very hot so that the DNA strands can break apart or denature. This allows those little bits of primers or those DNA strands to come in and prime or start the synthesis of new DNA. And this process is called annealing. So they're just basically sticking onto those strands of DNA. And then our polymerase can come in and they can use our nucleotides to add our bases to our new strand. And this happens in two different directions. So for every one strand we start with, we end up with two new strands. And so this is an exponential amplification of a target gene or a target portion of DNA. And this happens over many, many cycles, so 25 to 30 times. And what we end up is thousands or millions of copies of what our target that we're aiming for. And so for us, what we're really looking at is these variable regions of DNA. So I did mention that all of the bacteria have the 16S ribosome and the 16S ribosomal RNA gene. And so those genes contain these green or conserved regions. So those regions are present in all bacteria, no matter what type of bacteria. What determines the differences between bacteria types and species are contained in these variable regions. And there are different variable regions within that gene that you can target in this next gen sequencing. After the sequencing is completed, we can get some data, and some of the things that we might get are some of these really nice colorful graphs. And so what these are showing are the relative number, so how many bacteria might be in a sample, and what groups are there. So bacteria can be broken up into several different major families or groups. And this technique allows us to separate them. You can see in this bar graph here that there is this black portion on the top, and these are kind of the unknowns. So even though we were able to get some data from it, we still don't have anything to match it up. So we're still trying to resolve some of these unknowns in some of these samples. We can also see that there are differences. Now, what we're really interested in our lab is one, looking for what we call a shared core. So are there regions or bacterial communities that are present in all belugas that might be essential for beluga health? And on the same turn, we're also looking for different or unusual patterns in the bacterial communities that may indicate emerging pathogens or an infection. So those are some of the things that we are interested in. However, technology is always moving forward, and now we can apply new techniques such as metagenomics, which will tell us not only what groups are there, but it will tell us exactly who is there down to the species level. Using a technique called metatranscriptomics, we can not only tell you who's there, but what they're actually doing. So what's the function of these microbes when they are in these animals? So how do we apply this to beluga conservation in the wild? So belugas are circumpolar animals. They inhabit both the Arctic and subarctic regions. And there are currently 5 genetically distinct stocks of beluga whales in the US. And for most part, belugas are doing fine, and their populations are stable or even growing. However, there are critically endangered populations of beluga whales, and in the US, the Cook Inlet population is critically endangered, and there are estimated to be less than 270 individuals left. Despite several mitigation efforts, their numbers are still declining. But why? We are not sure. Well, there are many things that are impacting belugas. The area where belugas live is ground zero for climate change. We can see here from these pictures of the receding polar ice caps. And this is melting sea ice is opening up new shipping routes for an increasing vessel traffic. And it's also increasing oil and gas drilling and exploration. And so all of these stressors from their environment make the animals who live there more susceptible to new or emerging pathogens or re-emerging pathogens. So maybe things that these animals have never seen or haven't seen for generations. And this can be bad for the belugas. So just think of what we have been through in the last 18 months with the COVID-19 virus. That was a novel new virus that humans had never been exposed to, and it had some very bad consequences worldwide. And so these emerging pathogens can come in. They are opportunistic. They can take over the good bacteria. And this can lower the diversity. Diversity, or having a lot of different types of bacteria, has been associated with better or good health. So that could definitely have a negative impact on their health. And so in summary, by collecting microbiome data from healthy populations in the wild, including populations here at the aquarium and other zoos and aquariums, we can compare those with microbiome data that we'll hopefully get from these endangered populations like the Cook Inlet population, to determine what might be happening and if there really is that true core group of bacteria that is necessary for beluga health. And so microbiome analysis is truly a new and emerging field, and how much of a role the microbiome does play in beluga's overall health, reproduction, and survival remains to be seen. But that is something that we are working on, and I welcome you to stay tuned and follow back up with us to see what we find in the near future. And so with that, thank you so much. And I would love to take any questions, or if you have anything else you would love to discuss.
Great. Thank you, Dr. Driscoll. Really appreciate it. There weren't any questions that came up yet in the chat, but oh, it looks like we have a hand up. Is that you, Bob?
I'm just applauding the excellent talk.
Oh,
Thank
nice.
you very much.
I will join you in that. Thank you. Thank you.
I do have some questions if no one else does. I've got many questions, so, but
yeah, great. I do too. So go ahead, Amy.
Thank you. So one of the things going through my mind was like how the microbiomes from wild populations differ from the captive populations at Mystic, and like how much of a shared core they have, or how much of that difference they have.
Yeah, so they are in different environments. So we certainly definitely don't expect them to be the same. And even individuals in the same environment are going to be very different. But by using our whales here, we can really see how the microbiome is changing. Does it change seasonally? We can look at those various different hotspots that I mentioned, not just that respiratory microbiome, and that will help us begin to understand and put in perspective that data that we can collect in the wild. So if we collect a sample from a whale during the summer months out in Alaska, what would we hope to see? What are we seeing in the whales here at the aquarium? How does that change seasonally? That sort of thing. And by collecting more and more of these samples, we can kind of try to put that profile into context. Good question though.
Great, thank you. Because I was also wondering how it relates to the other microbiomes on like the skin or the other part.
Yeah, they are really different. Like I said, so certain bacteria, they do take advantage of those different niches. So they might be different pHs. They might have different nutrients that those microbes are exposed to. It is fascinating to me that they do share very little in common with the water that they swim and live in. So it is very distinct to their skin. And we have seen populations like in other aquariums, when animals are moved, their microbiomes might adapt on their skin to reflect the microbiomes of the animals that are living there. So they do change. Again, they change with illness, they can change seasonally. So there's a lot to kind of sort through. Yeah.
There was one that came up in the chat that says, is there a link or a paper with images and description of the snotbot?
Yes, there are a lot of snotbot studies. So the snotbot is one of these. I think the One our student is working on is called the blowalyzer. So stay tuned for that. I think we're going to get ready to patent that. But yes, there are a lot of snot bots. A lot of those, again, those are using those big whales where you can really get a really good sample either by putting that Petri dish or even an assay plate, attaching it to it and getting a lot of that genetic material.
Huh. Interesting.
So I think this is a fascinating example of using a charismatic megafauna, the beluga, which you can love in a lot of different ways, to getting to microbes, which many kids don't particularly appreciate, to, PCR. And it was a great description of PCR, but, oh, I have to know this to get to that because I care about this whale, like this transfer. Does anybody on in the audience here have they gone this whole gamut from from charismatic megafauna to PCR, or detailed laboratory? Does anybody have any experience with that?
Great question, Bob.
So maybe we should do this. This is a great one.
Yeah.
I'd say about microbiomes. I usually talk about stomachs, but whales.
Well, we use poop, so
Yeah.
you can go from poop
Yeah.
to penguins,
Yeah.
or penguins to poop.
Yeah, and I have done some high school classes where we got permission to collect seal scat and then process the seal scat, not to the microbiome level, but just to the what are these seals eating? Because this was a very basic, like, we're just gonna sieve poop, which was fascinating because at the time, the local fishermen were saying, well, they're eating the fish that we want, so they need to go. And we found that wasn't necessarily true.
Yeah,
And we certainly do it here, but we're fortunate, to have the resources that we do. And I was curious too, if there's high school teachers out in the audience, if they even have equipment that they can use in their schools, to look at that molecular level. And maybe talk about ways around that, because I know, microscopes it's a pretty expensive piece of equipment, but there's other tools out there using your phone, and adaptations you can buy for your phone that can help. So I was just curious if anybody teaches high school and can comment on that, ways around the expensive equipment to try to get to that interesting stuff.
I do know from high school science night that you can extract DNA from things like strawberries or spit. And so
That's right.
That's a good access point, right? Even if you then get to the snotbot paper or the studies, whatever, that there's a hands-on activity that doesn't take very long. That can connect the two.
Yeah, that's great. I know, and to your point, Bob, I hated science in high school, unfortunately, and that was because I didn't have very good teachers or inspiration, unfortunately, at that time. But man, when so I didn't even know much about the human body, and I had no interest in learning about the human body. I cared more about animals. So, but when I started to learn about a charismatic megafauna being a dolphin, in my first job in the career, I was like, well, I better learn about the human body. Otherwise, I can't appreciate the amazing adaptations this animal has as a mammal living in the ocean. So, your point is a really good one, that you can really use some of these types of things to inspire kids to look deeper. Another one that's come out recently was looking at discovering the existence of microplastics, in marine mammals that are in Antarctica that don't have any interaction with humans whatsoever. So, and it's a big eye opener, I think, for kids when they hear something like that and realize that, yeah, our impact is everywhere across the globe. And I think they found it in scat, didn't they?
Yeah.
Dr. Driscoll? Yeah, it was with sea lions, right?
Yeah, I think so. Yeah, they're finding it in whales too, I think now too. Yeah.
To go back to using some of these techniques in high school classrooms, I learned about a really cool resource yesterday called Lab Exchange. And so they have a bunch of like online ways that you can go in and do a lot of the PCR micropipetting types of experiences with students, and it'll lead you through it if you don't have that in your high schools.
Yeah, I think the online resources are just so amazing now to, if you can't do it, at least show it, visually through videos and stuff. Obviously, it's always good to do it yourself. But.
Yeah, that's it, Amy.
Is that the one? Okay, that's great. Yeah.
Oh, awesome.
Yeah, and I know
No, go ahead.
Oh, I was just gonna say, I think it was I'm trying to think the resource we used. I'll see if I can find it, but I know there's like adaptations you can attach to your phone that will make it pretty, and they were only like $30. I'll see if I can find it that can make it pretty powerful.
Are you talking to the microscope stuff?
Yeah.
Yeah, we did that for that pre-college class. Yeah, you can buy little microscope attachments for your smartphone for under $30 off of Amazon. Yeah, they do. And they took really great pictures. I was pleasantly surprised, actually.
We had a science cafe where Colleen Kavanaugh gave a little spiel about her gut microbiome stuff, and she said that the weight of the gut microbiome, the bacteria, is the same as the size, the weight of your brain. And that was a really interesting analogy, right? You go, oh, the brain's really important for like us, for thinking and being human and stuff, and your gut microbiome is the same importance. And you go, whoa, didn't really think about that, but yeah, all the plumbing and sort of has to. So that was one thing. And then she pulled out a little microcentrifuge tube full of some bacteria she was working on, like out of her purse. And so the fact that I remember that, right? Oh, I always travel around with some. Would you like some? She says, and she hands out these things. So I offer those as examples, but also a question, Maureen, do you interact with public audiences, obviously. Do you have some things that work great, or do you have some examples or situations where you would like some help from us, from the group here, thinking about how to bridge to an audience or something like that?
Yeah, always looking. We have done the extract the DNA from your cheek. Yeah, that's definitely a popular one, and it's a good visual. The kids are always amazed that you can see your own DNA. So that's always a fun one. I'm big with props, so I might always try to create something. But yeah, I don't carry around bacteria in my pocket most of the time. Maybe I should.
So, Dr. Driscoll, you mentioned that the shared core, and I think Amy was kind of getting to that earlier too. Like, do we really know yet what that shared core is? Or are you guys still trying to figure that out?
No, we don't. We in other data that other groups have shown and looked at, you can see where there are definitely differences, but actually saying like, okay, this is the core. But with these advanced techniques like the metagenomics, where we can read not just very small pieces of that gene, but larger pieces, and really get down to like the nitty gritty, I think that that's gonna be the real telling thing, because there are just very general groups, and there can be, beneficial bacteria in those groups as well as pathogens. So just because you might see like one group, it doesn't necessarily like throw off an alarm bell that there might be something going on with that whale. But we're hoping that with these cook inlet belugas and being able to collect some blow with these drones and, some of the drone technology other groups are working and getting some of those samples, that maybe something will stick out from those guys, because we know that there's something going on there. Yeah.
Right. It was interesting to hear about the, the microbiomes of the ocean water itself. I imagine there's a lot of diversity there too, depending on where in the world you're collecting water from. And so do you find correlations of what's present in the water versus, I know you said it was like totally opposite of what was found on the skin, I think at one point, but.
There is some overlap, but again, and this is something that I know they look at Woods Hole, and they're looking at a lot. And, is that stress from that environment weakening that skin microbiome and allowing some of those microbes that are in the ocean just swimming around and being able to come in and maybe cause infection or some other types of disease or problem.
Yeah.
So I'm really curious about this shared core now, and I've got tons of questions, but so have you compared, does the shared core just between belugas? Does it correlate with other whales? Would it are there some shared core between human microbiomes, like respiratory microbiomes?
It looks like it's definitely species dependent, and it's definitely swayed by the environment. So I think that the environment definitely has a large role to play in it. And so that, puts a little caveat about comparing things from different populations. So we're still trying to get some wild samples for this. So that's kind of like one of our ongoing goals. And with the whole COVID and not being able to get out there and get any field samples, but that is the top priority. So we have a lot from, some belugas under human care and some others, a few from some wild, stable, healthy populations, but just trying to get more from different parts. There's also an endangered population in the St. Lawrence estuary. So those belugas, they are endangered. Pretty sure that it's because of all of the toxins and pollutants that are pumped into that body of water, all of the shipping, a lot of those animals have gastric cancers and stuff like that. So getting those where we kind of have a little bit better idea why those population is declining, whereas like the Cook Inlet, after mitigating, pollution and shipping and stuff, it doesn't really seem to be tipping the scale in their favor.
So I'm going to put my teacher hat on, and it's a pretty small group. I'm wondering if we can go around the room and just introduce ourselves and see if there's some questions or thoughts that we have. My name is Bob Chen. I'm an oceanographer, and done a lot of education across different age groups from, I don't know, first grade through gray. Amy?
Yeah, I'm Amy Gollenberg. Oh, sorry, Amy Gollenberg.
No worries, there's you go for it.
Oh, sorry, I was looking at two Amy's at the same time.
Okay, I'm Amy Bonanno. I'm program manager of NEOSEC, and I work with Bob Chen at UMass Boston. And I've done marine research and marine education.
And I'm Amy Gollenberg. I'm our director of school programs and have done mostly informal education, but I've worked with a lot of formal education as well. So anything from preschool, as you said, to, all the way up through the later stages in life, the silver linings. And yeah, it's, one thing I just wanted to add to this is, when you were talking about megafauna earlier and the inspiration, we often talk with the kids about the fact that especially marine mammals are a good indicator of the health of the overall health of the ocean. And that's what all of this ties into. So, they can really, their presence and what their state is, what their healthy state is or not. And that's why this research is so important, because it can really indicate the health of the ecosystem they're in. So.
Definitely.
I guess I'll go next. I'm Megan. I'm the conference chair, but I also work for New England Science and Sailing. I have done both formal and informal education. Mostly though, I prefer the informal, and I'm the program director here. So I have taught anywhere from 3s and 4s all the way up, like Amy G, all the way up till, you know, the older version of people. But, you know, I really love bringing this sort of science to the littlest ones. You know, we just did ROVs with 4-year-olds, which was the most eye-opening and wonderful experience I have ever had. And that's my favorite part, is to really make big science to little people.
Thanks. Paul?
Oops. Sorry, I have to give Chrome permission. I'm Paul Berube. I'm a research scientist at MIT. I thought it was a great talk. And in the, I was also thinking in terms of like the PCR and giving access to high schools. I was kind of looking up to see if MIT had anything like along those lines, but I was wondering if there's any outreach to sequencing centers, even like local universities that, you know, if someone's doing a sequencing run for 16S, it's probably not that much more expensive just to throw an extra, you know, an extra sample on it, or extra couple of samples from a participating high school. So I'm just wondering if that's an avenue to get data, and you can put it through a normal like computational pipeline and get back some neat like pie charts and stuff that the students might be interested in.
That's a great connection.
Yeah, I know that for the Yukon, for example, you can send them your sample and they'll extract the DNA and they'll do it right through the whole thing, you know, for under $40 a sample from start to finish, which isn't too bad. And then other places like URI, I think you can send them your DNA and they'll run the PCR for you. So I think there are some universities that have those core labs that you can send things to. And then there's some other private companies too that like Eurofins that are, and everything is very inexpensive now too. If you have that DNA, I think you can just send it right out and get them to do it for a few dollars probably.
Awesome. Molly? Are you there, Molly? Christopher? Julie?
Hi, I'm a high school science teacher in Mid-Coast, Maine. So whenever we get opportunity to utilize data and take big data and be able to chunk it down, I try to do that for my students. Do we have access to, I heard was DNA sampling of seal poop. Is there kind of like a group area where you can gather that data or get it for your students to use in the classroom?
I'm not sure. I'm sure that there is data. I know that's one of the things we're doing that as well, is looking at, you know, seal poop and poop from everything actually. But yeah,
Right.
that is a good question. There are groups of people that might be something that you can even get through. Maybe like NOAA has databases that they kind of dump things in. So that might be, I don't know if it would be maybe microbial data, but that might be a place to start. I know that there are a lot of those public data kind of dumps that groups put things into.
And Julie, Amy G and I have a friend who worked very closely with the seal scat studies, Amy Ferlin. And I can reach out to her and see if I can get an idea of where her data went. I'm not sure if she did seal scat or just seal populations. So in Long Island Sound, I should say specifically. So I can't necessarily help you in Maine, but I can help you down this area. Yeah.
Right. Thank you so much.
Julie, I was so there's something called the Magnify service. It's run by the European Bioinformatics like clearinghouse. Like the US and the Europeans have their own, we have NCBI versus EBI in the EBL over in Europe. But Magnify is where you can find all these metagenomes, and they're, like, I've been focusing on like the open ocean ones, but there's probably ones from any sort of like sample. And they've already run everything through their pipeline, looking at annotating things for functions and 16S. So that might be useful. I'll drop the link in the chat. Maybe it'll be useful.
Thank you. I have to say, I also teach chemistry. So the biology side of my brain hasn't been used in a couple of decades. I'm good on the chemistry. Julia Huber, I was on. But when we start talking about DNA, I'm like, oh my God, this was all invented after my last major biology course. So really trying to keep up on it and make myself sound semi-intelligent for my students so I can send them in the world.
I think this piece is absolutely critical, connecting, in the classroom, teachers with scientists, so that when you have a question, you can get not like all of science, right, but the one resource that you need that
Right.
you use. And so
Right.
definitely recommend the networking sessions this afternoon. Also, that's a key part of the NEOSEC conference.
Wonderful. Thank you very much for that. Appreciate it.
What else do we have? We did to everybody. And it's interesting to hear you say that, Julie, because when I was in college, organic chemistry, I struggled and struggled and struggled so much. And but then I took microbiology afterwards, and I was like, you know what? If I had taken this first, and then I would have totally appreciated organic chemistry so much more. Like, why did you set it up this way?
The type of chemistry I do is a lot of environmental chemistry from my background, because I did informal education in a previous career. And so, applying it, making it real, not just a Sudoku puzzle, is the group that I work with. And I have students who make more in a summer lobstering than I do, in 2 or 3 jobs. So, getting them to understand their community, and when we talked about the seal poop and were they actually eating what the fishermen say they were eating, I think is real connection to the students that I have that are working on their own boats. Also, I'm heading over to the Atlantic Shark Conservancy tomorrow. So I really want to look at how do we look at different poop who's available. The eDNA piece to it is really kind of helping my students understand the bigger picture.
That's fantastic.
Yeah.
See, Chris is plugged in. Do you want to say hello, Chris? Or
Yeah.
Yeah, I'm here. Hello, I'm Chris. So I work at the Gulf of Maine Research Institute in Portland, Maine, and I'm a lab educator. So we're working primarily with 5th and 6th graders, and getting as many of the 5th and 6th grade students in Maine to our doors as we can, which thankfully we're able to do this year. Yeah.
So white beluga whales are pretty engaging.
Oh yeah.
Excellent. Well, it's 11:51. I think it's been really fantastic. Is there anything else you wanted to add, Maureen, to before we depart, or anyone else for that matter?
Just that I hope to come back and give some better answers maybe next time, and hopefully we'll start really. I know that's like our top goal of next year is really to focus on these microbiomes and realizing how important a role they are playing in the health of these animals. So,
very true. Thank you. Thank you. Well, we look forward to the results.
All right. Thank you, everybody.
And I
Thank
want
you,
to
Maureen.
encourage everybody
Thank
to
you.
come to trivia.
Yes, trivia at noon.
10 minutes.
Yes, absolutely.
Thank you, everybody.
Thank you all.